What instrument does Yo-Yo Ma play? Cello, Elasmites. Welcome to the show that tunes your strings and plays you like a cello. I, your host, Sam McNeely, will be orchestrating today's episode like always, and I'm going to be honest, for this one, I was pretty starstruck. When I emailed today's guest, I literally told myself, no way I'll even get a response. Like, they can't possibly have time for me. But clearly they did respond and had time to talk to me because, this episode exists, so. Anyways, let's get rolling with some clerical business before we meet our great white shark expert. First, did you know I tell a little secret or weekly update about what I'm up to after the credits at the end of each episode? I try to give a little hint into what my chaotic life is like, so make sure you stick around long enough to hear that because I try to give advice or real-time updates on just life, but also with my experiences in marine science. Anyways, thanks for listening today, whether this is your first time or you've been here before. I also want to give a huge thank you to all you Patreon donors who keep this show going continuously by giving at patreon.com/elasmospod. If you're thinking about supporting this podcast, please check out that Patreon page to see what additional benefits you can receive by becoming an Elasmite donor. Do it for the sharks. So that's patreon.com/elasmospod. Also, thank you to those who rate and review this podcast. And remember, if you rate and review this podcast on whatever platform you're listening to it on, and send that rating and review to me at elasmospod@gmail.com, you'll be entered into a raffle to win a prize. Yeah, so good to it. And also, it makes this podcast known to get out to more people and to also bring more people to want to be on the show. So you want to hear great people? Rate and review this podcast. Here's a hint. It works. And also, if you share this podcast on social media, you'll get 2 extra entries into the drawing. So rate, review, share on social media, and email me at elasmospod@gmail.com. You can find the Elasmos Podcast on Instagram and Twitter at elasmospod, and our Facebook page and YouTube channel are both called the Elasmos Podcast, A Shark's Universe. Our website is elasmospod.com, where you can find links to all of these media outlets. And lastly, don't forget to send in your listeners' adventure stories to me at elasmospod@gmail.com for our listeners' episode. And of course, to encourage ecotourism, which we love. Go explore nature. Okay, let's do this thing. Well, let's do the first part of this thing. I chatted our guest up so much that I'm having to actually make it a 2-parter. Guys, I just couldn't stop asking questions. My curiosity just ran like a mouse on a wheel. Which, to be fair, is kind of what my brain is like most of the time, so kind of par for the course. But anyways, our guest this week is a famous one. Like, if you've ever watched Shark Week, you've most likely seen this guy. That's right. By day, he's a senior fisheries biologist that specializes in great white shark research with the Massachusetts Division of Marine Fisheries, where he heads up the Massachusetts Shark Research Program. He's also a guest investigator at Woods Hole Oceanographic Institute. He got his master's degree from the University of Rhode Island and a PhD from Boston University. He's been featured on National Geographic, Discovery, and ESPN, and he even has his own IMDb page, which I just literally found out after doing a quick Google search of his name. So when people are making movies about sharks, they go to him to make sure their stuff is fairly accurate. Not the most accurate because shark tornadoes don't really happen, but maybe making sure they know how a shark works. I don't know. He's helped out with movies and that's badass enough for me, so. And also, this is like a summed up version of his intro because, the list just keeps going. And in this first part, we talked about the importance of volunteering, shark vertebrae, the beauty of being a non-traditional student, which I happily classify myself as. Life advice for everyone, not just shark lovers. You heard that right. If you're not a shark lover yet, stick around. This guest will convince you. We also talked about what happens to a fish after it's caught and released, and of course, why the health of the ocean depends on sharks. So Elasmites, get as excited as I was to meet great white shark expert, Dr. Greg Skomal. This is episode 10, part 1.
Cool. All right. We are rolling.
Awesome. Well, let's get down to it. Well, actually, before we get down to it, I really just want to say thank you so much for doing this. It means so much. You've been such a role model for me in the world of marine biology and marine science. I just finished my undergraduate degree in marine biology at UNC Wilmington.
That's great.
So I've really looked up to you throughout my undergraduate career. And even before then, when I was just watching Shark Week and everything, just watching you was really I was putting myself in that position. So you've really been a role model for me, and it's almost surreal meeting you, even virtually. But I just really want to thank you so much. And actually, during the pandemic, I've been able to listen in on some of your talks. And as such a note about it, I was really geeking out about you giving a talk, and so I signed up immediately. I was like, of course. And I got my parents in on it too, and we all just made a night of it. It was like our movie night, but instead, we watched you and another scientist talk about great white sharks. So it was really cool. And honestly, it's kind of like a dream come true meeting you. So I just can't thank you enough for doing this with me.
Well, I'm honored. Thank you very much, Sam. I hope you pursue it, and I hope you succeed. I really do.
Thank you. That really means a lot.
You're welcome.
I never really stopped fanboying over Greg, and to be honest, I don't think I ever will. He's just that cool to me. But I eventually got to my question of where he's from and how he grew up. Don't worry, y'all. I got there. It took me a while, but I did it.
Sure. Yeah. I'm originally from Southern Connecticut. I was born in Stamford. Which is a stone's throw from New York City. And then when I was 8 years old, we moved a little bit to the east to Fairfield, in Fairfield County, and pretty much grew up there. Went to high school there, and then when I graduated, I moved on to Rhode Island, where I went to the University of Rhode Island for my undergraduate degree, and then stuck around for a few more years for a master's degree. What's interesting about growing up, now I grew up in the 70s, so, it's a long time ago. And so, we didn't have any of the technology we have right now, but I grew up not too far from Long Island Sound, which is the body of water that separates Connecticut from Long Island.
He must have known I was geographically illiterate. Now I know. Look at us, learning geography together. Who would have expected that on a shark podcast?
But remarkably, I didn't spend a lot of time down in the Sound. Back in those days, it wasn't all that exciting to be, underwater or in the water in Long Island Sound. It's changed a lot since then. So a lot of my early experiences with marine biology and sharks were all, virtual, and all through the, television shows that were on and movies like Jaws and, Blue Water, White Death and, other films. And so that really was most of my inspiration, seeing guys like Jacques Cousteau go underwater and study coral reefs and sharks. And it was all about that. So I had aquariums as a kid. I loved to have fresh water, and then I eventually went to saltwater aquariums. And then I was pretty lucky to get to travel down to the Caribbean too, in the mid to late 70s. And that's really what inspired me to become a marine biologist.
Yeah, the water's so crystal clear, and I can only imagine what it was like before some of the diseases hit, like the corals and stuff. I mean, it must have been just absolutely pristine.
Yeah.
Like an underwater wonder world.
Oh, it's fantastic. Just to be able to, I was used to buying fish for an aquarium, but then to go swim with them, and not see them in a glass box, but to see them in a natural coral reef ecosystem, that was like, I got to do this for a living,
That's exactly how I feel. It's just so much different. So sounds like we're on the same wavelength.
Great. That's great.
Now, so what was that first moment that you really became interested in science that was like, okay, this is what I want to do. This is what I want to keep doing?
Well, I think it was pretty much, realizing that, the stuff that Jacques Cousteau was doing and, that he was sending home a lot of really pretty pictures to our TV set
Right.
But it seemed to some extent he was conducting some level of science. But the truth of the matter is, in order to be a marine biologist and get those experiences of interacting with fish and being underwater and scuba diving, you had to be a scientist. And so I was thinking early on in high school that, to become a scientist, I had to, take science classes and see if they were interesting to me, which they were. But in 1975, a popular film by the name of Jaws came out. And one of the characters in that film was a guy named Matt Hooper, of course, and he was a shark biologist. And that really, hit home for me in terms of, wow, this guy is studying sharks and he's a scientist and he sounds like a scientist. And, I need to become a marine biologist. And so that was another inspiration for me, that really odd little character in the book and in the film that said, wow, this guy knows about these animals. And, being a marine biologist isn't just swimming with fish. It was trying to learn something about their biology, and for me, not only learning about their biology, but producing the kinds of information that ultimately leads to sustainable fisheries management is what really drives me.
I mean, that's interesting. That's something that I've talked with a bunch of other people about too, is that, the general consensus of Jaws seems to be that it's fear-producing. It's this fear-mongering man-eater shark.
Yeah.
But then the people that I've talked to on this podcast that have mentioned Jaws, they've all been inspired by it because they saw Matt Hooper and they were like, oh my gosh, Matt Hooper is doing this for a job. He's doing this to try to understand the shark. And so it's really funny and interesting to see how there's kind of like a back and forth between the two ideas of sharks are these terrifying creatures, I need to stay far away, to sharks are really interesting now and I want to learn more about them and kind of change the perception that this movie has created and learn more about them as well.
Yeah, it is. And, back in the old days, marine biologists were not necessarily expected to spend a lot of time sharing their information with the public. So we were, the scientific community was a little bit to blame for that perception and not battling that perception. I think perceptions have changed over the years, but I think with better communication, with better tools and gadgets that we use and social media and the internet, many scientists, most I know, freely share that information. And there are a lot of great new vectors to do that, that allow us to do that, so we can interact. Like, for example, this podcast, right? It allows me to share scientific information with the public and set the record straight about these animals. And so I think that's resulted in more fascination as opposed to fear when it comes to sharks.
Oh, yeah, absolutely. And that's what I love that's grown so much is science communication and how it's used to change perceptions. And honestly, that's the purpose of this podcast, is to change the perception from them being these fear-producing animals to animals that deserve our respect and, ultimately, our love, but mostly respect.
And which is, as far as I'm concerned, fantastic,
Thank you. I'm trying out here. I'm trying. And so going back to your education, you said you did your master's at the University of Rhode Island?
Yeah, I did my undergrad, and what was really cool, University of Rhode Island has the Narragansett Bay campus. It has the Graduate School of Oceanography. It has Marine Biology Department. It didn't have marine biology back then, but, I had access to all kinds of scientists and experiences and coursework through URI. But I also, right down the road next to the Narragansett Bay campus was this National Marine Fisheries Service federal laboratory. And in that lab was a particular program that was the cooperative shark tagging program. And so I began to volunteer for that program when I was a senior, my last year as an undergraduate at URI. And so I was volunteering, and I can't say enough about volunteering. It's important. You get great experience. It also put me in the right position when they were hiring a technician during my last year at URI they asked me if I would like the job. And so I started working for the cooperative shark tagging program, which not only was tagging sharks, but studying sharks relative to all aspects of their natural history.
Oh, awesome.
But in order for me to keep working for the program, I had to stay a student. And so when I graduated from URI, I just rolled that over into a master's and stayed on, and then ultimately got my master's degree, but doing it on sharks. And so with the cooperative shark tagging program. So it really put me in a great position to not only close out my undergrad, but to get that master's degree as well.
Now that's really cool. So you were able to link your master's work with the program you were already helping out with.
Exactly. And I learned a lot from those scientists, and to this day, they're good friends and they're mentors of mine.
Oh, I love that. That's awesome. And yeah, really, volunteering is just such a stepping stone, especially within the field of marine science.
Yeah.
Clearly for you, it's had an impact. I mean, you started out volunteering with this program that led to a master's, I think that's incredible. And it seems like a lot of programs do that too. But so what was your master's thesis focused on?
Well, it was determining the age and the growth rates of the blue shark. And the blue shark is by far the most abundant large pelagic shark we have off our coast here. Probably in the world, actually. And no one had ever done a study that would examine, size at age, longevity, and their growth rate. And so I did that at the university, coupled with my experience at the lab. So they gave me access to all my samples. And back in those days, the samples were shark vertebrae. So they were backbones from blue sharks that had been collected at various places, including fishing tournaments and commercial fish houses and such. So I was, in essence, counting rings on the vertebrae of those sharks to determine their age, and also trying to determine, the frequency at which rings are laid down in the vertebrae, because they're not trees, and we don't know if they're annual or not. So that was my entire master's thesis, and it was an incredible experience.
Oh, I can imagine. That's got to be so fun. And so interesting, too. And now you said they're not like tree rings, but is there a quote-unquote growing season similar to tree rings?
Yeah, what we found out with the blue shark is, the rings tend to be annual. All right. So they do lay down a check of a growth band, and it probably coincides with the time of year that they're not growing as fast, which is likely during the colder months, or perhaps during long periods of migration. And so we determined that blue sharks, for the most part, live somewhere between 15 and 20 years as their maximum longevity, maybe a little bit longer. In their early years, they grow about a foot a year, and they get to be about 12 feet long, but their growth rate slows after they reach maturity, which is somewhere between 4 and 5 years old.
Oh, okay.
And so we ultimately published that information in a peer-reviewed journal, and it is still used as the primary growth curve for the blue shark for most stock assessments.
Now, so is that methodology used for most shark species, or just the blue shark?
It is the primary method that is used to age sharks, and has been for probably 100 years.
Wow.
More recent work I've done has involved the white shark, but along with a colleague of mine, Lisa Natanson, who does a lot of growth work with sharks, we recently published a paper a couple of years ago that really calls into question the use of backbones and rings as an aging method for shark species. And so we kind of threw a wrench in the works and are shaking up the standard paradigm. We used a couple of new techniques to demonstrate that the rings may not necessarily coincide with time as much as they coincide with the growth of the animal and the need for stability within the backbone to support the movement of the animal. And so people still use backbones, but we approach it more delicately and with corroborating methods to demonstrate those growth curves are real.
Wow. That sounds like a difficult process. And it sounds super detailed.
There are lots of details and lots of curveballs, but, it's really the only method we've got. It'd be one thing if I could throw a blue shark in an aquarium and watch it grow for 20 years, but that's not possible,
Yeah, if only. That's a bit more difficult to do.
Yeah, a lot harder.
Yeah. And now you went to Boston University for your PhD, didn't you?
Yeah, but another lesson for folks out there who are interested in this field, they may not necessarily have the time or the money or the energy to go, get these advanced degrees. So what I did in 1987, I left the lab I was working at in Rhode Island and took a job with the state agency in Massachusetts, the Massachusetts Division of Marine Fisheries, who I have been working with now for 34 years. And so there were a couple of reasons I took the job. It was because, my degree was ending in Rhode Island and I had to move on eventually. I was no longer a student, but also the job was presented to me as being located on Martha's Vineyard Island.
Ooh, that's nice.
And I'd never really lived out there. And I thought, I love islands. I loved islands all as a kid. And I figured it'd be really cool to work on an island, and not only an island, but an island where Jaws was filmed, and I was also given great latitude to start my own job and to sort out what my job would be. And so I moved in 1987 to the Vineyard, and it wasn't until really 10 years later that I decided to go back and get a PhD.
Oh, wow.
And so I took 10 years off from school, which was great. It allowed me to develop my professional career.
Mm-hmm.
I started the shark program for the state of Massachusetts, and we were doing all kinds of research, not only locally, but on a global scale with a variety of researchers. But I felt like I needed to continue to grow. I'm one of those people. So I bumped into a friend of mine who was a professor at Boston University. And in those days, Boston University had a marine program that was located in Woods Hole, Massachusetts. And Woods Hole is right across from the Vineyard. So he accepted me into that program. They gave me a teaching fellowship, and I was able to commute right over to Woods Hole from the Vineyard and work on my PhD while keeping my job and my normal duties. And so it was really another one of these incredibly ideal scenarios where everything just clicked.
Yeah, no kidding.
But I'm one of these people that if you approach life with great optimism and energy and you wrap your arms around it and you commit yourself, things will fall in line for you. And I've been very fortunate.
Yeah, that's awesome. I mean, I was just about to say that sounds like the stars just kind of aligned for you to work with sharks and to keep pursuing further degrees.
Yeah. I tell you, they really did. I had to work hard, but that's what you got to do.
Of course.
There's no handouts. So you got to work. You got to work hard, and that's what it's all about. You got to apply yourself. You have to have the right attitude, the right approach.
Mm-hmm.
You got to think outside the box, and you have to really sometimes put yourself in uncomfortable positions. But that's how you advance yourself.
I believe that too, and that's some dedication to work for 10 years and then decide you want to go back to school.
Yeah.
I feel like not many people would want to go back and relive that again. And you said you were able to keep your job while you worked on your PhD. Your PhD must have been related somehow. I don't know how you'd have the time otherwise. But maybe that's just my chaotic mind getting stressed about all the things that I would have to do.
Yeah, and that was one of the incentives, really. Just like with my master's degree, I was working at a program. They offered me this project. I turned it into a master's. At the State Division of Marine Fisheries, I was working on a very specific project that was trying to examine, post-release survivorship and behavior for fishes, big fish, including sharks and tunas and marlin, that were exposed to the stress of being captured.
Interesting.
All too often, state and federal agencies mandate the release of fish. Anyone who goes fishing in freshwater or saltwater, for striped bass or bluefish, that you can't keep fish that are, under a certain size.
Mm-hmm.
And so we're basically telling you got to throw that fish back. And that applies to most species, so blue sharks, makos, white sharks, bluefin tuna, white marlin, we're telling people you got to let them go. the question that hadn't been answered is, what happens to these fish when you're struggling so much on a rod and reel line, a hook? And, do they survive? Because if they all die, there's no point in telling people to release them. And so we have to think of a better way to save fish. And so my interests were really big on, and still are, what happens to these fish when they are exposed to that level of physical trauma and physiological stress. And so that became the subject of my PhD dissertation.
Oh, awesome.
Looking at what happens to bluefin tuna, yellowfin tuna, white marlin, blue sharks, makos.
Yeah.
When somebody catches one, lets it go. Does it live? And how does it behave? And so I basically rolled that over into a PhD.
That's such an important question to ask. And to me, it sounds like a really difficult one. I guess first off, how did you even track the sharks after they were released? Was it just through some sort of tag?
Yeah, exactly. I basically had a two-pronged approach. One of it was every fish that we captured, we would sample its blood, and for anybody who ever goes to a doctor or takes their pet to a veterinarian, you can learn a lot about the health of that animal through a blood sample. And so we'd get a snapshot of that animal's health and to what extent it was stressed at the point of capture. And then we would affix all kinds of fancy tags, and that's where a lot of new tagging experience came to me. Using acoustic tags, using satellite-based technology to see how the animal actually behaved after we let it go. And so we coupled the stress that we quantified in the blood with the post-release behavior to get a sense of what the impacts were on that particular fish.
Oh, fascinating. And I was reading one of your papers that was focused on the primary, secondary, and tertiary effects of anthropogenic human stressors like catch and release. Don't worry, I'll put a link in the show notes for everyone to go read this great paper. So could you explain what the differences between primary, secondary, and tertiary stress effects are on a fish?
Sure. Well, any kind of stressful event or stressor, right? And this applies to almost any animal, it applies to us. It applies to your cat. It applies to fish in the ocean. There's a sequence of events that cascade when we get into a stressful situation. In the typical case of fish, it's basically the fight or flight response. So the first initial, perception of stress leads to what we call primary effects. And those are typically hormonal,
Okay.
Adrenaline, noradrenaline, epinephrine, cortisol, all of these are released. These are hormones that are released during the stressful event that lead to what we call secondary effects. And those secondary effects could be alterations in other aspects of your blood chemistry. So it could be a buildup of carbon dioxide, for example, or it could, if you're forced to run away from a stressful event, you're going to have the buildup of lactic acid because you're running like crazy, and fish are struggling like crazy to get away. So they'll get a buildup of lactic acid, which could also lead to changes in internal pH of your blood as well as your tissues. So your blood pH will plummet, because of lactic acid and the buildup of carbon dioxide. So this is called acidosis that occurs, and so those are the secondary effects of stress. Your body is also saying, I need energy to deal with this. And so it's releasing, glucose or glycogen from your liver or other energy stores, so your body's basically saying, I've got to respond to this stressful event and I need energy to do that. But at the same time, I might shut down other systems. So you might shut down your immune system because the immune system is incredibly costly. It's energetically costly. So your body is going through all these changes with its primary goal being, I need to survive this stress. That ultimately can lead to what we call third level or tertiary effects. Is the stressful event so bad that it could lead to long-term chronic stress or events that could alter your ability to reproduce, your ability to grow naturally. Your immune system may become compromised to the extent that disease or pathogens can take over. So those are the long-term effects of stress. So in a couple of book chapters that I've written and papers, we talk about how this happens in sharks, and it's very similar to other animals, and ultimately does it compromise the long-term survivorship of not only that individual of the population.
Oh, I gotcha. So it's just like cascading effects down the line of however, I guess, intense the stress may be or how long it may last. It could just have longer lasting effects. And how is that measurable? How do you measure tertiary effects? Because with that, I feel like you'd need to be able to see the shark through its entire life. And I mean, that just sounds really difficult, I'm going to be honest. Because can't some great whites live, I don't know, crazy amounts of time? Like 50 years or so? Of course I just looked this up. This is so cool. Okay, so past research suggested that great whites could live up to about 23 years. That's a good amount of time. That's about how old I am. Right now, I'd beat out great whites by 1 year. But recent research, done by none other than yours truly, Dr. Greg Skomal, and also a few others, of course, using radiocarbon dating of vertebrae, found that one great white shark lived to be 73 years old. 73! That's nuts. That's, how many times is that? 1, 2. That's over 3 times as long as they thought. So, great whites basically have the same lifespan as humans. Okay, let's get back on track. How do researchers measure tertiary effects in great white sharks, since it kind of takes seeing their entire life pan out? Here he is.
It's really difficult. So I can measure the primary effects, and I can look at the secondary effects through blood chemistry. That's easy. I can measure pH. I can measure hormone levels. When it comes to the tertiary effects, all we can really do is, through sampling fish in the wild, look at incidence of disease, for example. So this is a little bit easier on fishes in captivity. There are many fishes in captivity that develop diseases, and that's likely due to chronic stress and the exposure to pathogens. All right. So to some extent, we can look at growth rates in captivity. We can look at reproductive success in captivity and see what impacts that stressor of captivity has on those animals. But in the wild, it's far more difficult.
Yeah.
Far more difficult. In essence, to capture a fish that, let's say, has been exposed to, capture in a gill net, and so you measure the blood in that fish and then you tag it and you let it go. Seeing what that fish does over the course of the next decade is going to be almost impossible.
Yeah.
Almost impossible. We can put a conventional tag on that fish or an acoustic tag on that fish and see if it continues to show up in its normal areas, see if it exhibits what we consider to be normal behavior. But that typically spans months to maybe a year or two. To get past that's really hard. And so, I'm thinking that the technology might not exist right now to do that. But certainly when you're a marine scientist doing what I do, Sam, you may have the tools that allow you to start to answer those questions.
Is this Greg trying to get me or you to join him and answer these questions?
Join me. Together we can rule the galaxy as father and son.
Okay, maybe not as father and son, but you get the idea. I feel like this would be a great area of study for aquariums because they've already caught the animals. They've, in a way, released them by putting them into a bigger tank for them to move around, and probably for the rest of their life. I mean, I know some aquariums keep animals for the rest of their life because they're either injured or for whatever reason. So I would think that aquariums have a really great opportunity to kind of use that to their advantage and try to analyze what these tertiary effects look like.
In many cases, absolutely. Reintroduction back into the wild is becoming something that's not happening a lot anymore. So you may have that animal, for the rest of its life. And of course, it's in the interest of those aquariums and zoos to have a healthy animal. So they are very, very focused on maintaining those critters in captivity, and keeping them incredibly healthy.
Mm-hmm.
And so they're doing quite a bit of work along those lines. And that's why they have veterinarians on staff and they're very cognizant of animal welfare.
And relating it back to your expertise, your specialty, how do great white sharks respond to catch and release, especially in regards to those primary and secondary stress effects?
Well, we've been able to quantify the stress response in white sharks during capture events. And I can tell you their blood chemistry becomes disrupted like it does in every other shark we've ever captured. So you get this cascade of primary effects.
Gotcha.
You get dramatic drops in blood pH.
Mm-hmm.
And we know that in most cases, those animals, when they are let go, they survive. The part we have really, again, going back to the previous conversation, we don't know what the long-term impacts are on those animals in terms of growth rates and reproductive success and bioenergetics. So one of the things that we do is when we're tagging white sharks off the coast of Cape Cod, and we've tagged over 200, we try to use the least invasive approach to doing that. And so we're not capturing them. We're not hooking them. We're not exposing them to capture stress. We're not handling them directly. We're not lifting them, out of the water or strapping them to the side of a boat. We want to be as cognizant of, the stress to the extent that we're minimizing it as much as possible. And so we use a different approach to tagging where we're actually locating the shark while it's hunting in shallow water and then going up to it and just placing the tag at the base of the dorsal fin, which is thought to be the least invasive technique, using a small intramuscular dart that goes under the skin into the base of the fin. And we think that way we're not going to impact its behavior.
And is that tag just a tracker, or can it also measure different compounds in the blood, different chemicals or hormones?
Yeah, those are the different kinds of tags that we're using to study movement ecology and behavior. Back in the days when we were catching white sharks and taking blood, that was the focus of that study. We've moved away from that. We're trying now to study the big picture and the small picture. What are these animals doing in time and space? And so in order to do that, we want to use a very, very low invasive approach. Handle them delicately.
Oh, yeah, absolutely. And when I've seen y'all on Shark Week, I've seen you standing off the bow, off the front of the boat, and you've got this spear-like shot that you essentially just jab into the shark's dorsal fin. And, it doesn't even seem like they're affected. When I've seen the videos, they just don't even seem fazed by it. If you haven't seen a video of this, don't worry, I've got a couple links that I'll put in the show notes of Greg actually tagging and tracking sharks. They're really interesting, but they're also just, it's just wild. So what he does, he basically is on like a 20-foot boat, small boat, right? And then there's a plank just hanging off the front or the bow of the boat.
Ahoy, children, I'm back, because it's time for you to walk the plank.
And it has rails around it, so don't worry, he's not going to fall off. But he walks along the plank to the end of the platform where he stands with a long pole, and that's when he prepares the long pole to try to tag the shark. And once they've tracked down the shark, he's looking over the side, and he uses that pole to essentially just thrust a dart into the shark's dorsal fin. It looks so invigorating. Oh, I really want to do it someday. I really want to do it. I've got a couple links in the show notes, so go watch those. Alright, let's get back to it.
Yeah, we get a wide range of reactions. Many of them don't even seem to care, which is
Really?
good because, you don't want a crazy reaction because it could lead to tell you that you're impacting the animal. But, some of them are startled. You're talking about a 10, a 12, a 13-foot, 14-foot shark that basically nobody messes with, right? And then for me to sneak up on it and then, jab it in the dorsal fin, I think it's mostly a startle response where it's like, what was that? Who dared do that to me? But, they'll dart off and then they'll settle out and they'll be fine.
Okay. Now, every time I see you or someone else up on that platform, up on that little plank, ready to plant that tag, I just imagine myself in that position, and that gets my heart rate up. But do you remember that first time that you did it and what that felt like?
Yeah. Oh, yeah. So, I hadn't done white sharks until probably 2014.
Really?
Well, I shouldn't say that. I did tag some white sharks in Mexico in 2013. But I had been tagging a lot of other shark species, blue sharks, makos, threshers,
Oh, okay.
a number of shark species using the same technique where the shark is swimming and you place the tag at the base of the dorsal fin. So I had a lot of experience, at least using the basic methodology, tagging pole, tagging needle, intramuscular dart, dorsal fin. So when it came time to do it from the pulpit of the boat, while the boat's moving, while the shark is moving, I have to admit, I had some anxiety, but at least I had some experience and knew how to do it.
Right.
And so, but I was incredibly hesitant to pull the trigger because you want to make sure you do it right. You don't want to tag the shark in the wrong place and impact its behavior. So, I'm getting much better at it, over the last, half a dozen years. We've now tagged over 200, 230, using this technique.
Gosh.
So it's a lot easier for me and I can do up to 5 or 6 in a day. Whereas early on, I'd be lucky to get 1 in a day because I was so cautious
Wow.
and careful and delicate. And so now, I don't have as much anxiety as I used to, but you do have to pick the right time, and it's all depends on, the conditions that you're working under, the conditions of the water and, the visibility in the water and what the shark is doing.
Right.
And where you are and how you're behaving relative to the shark and the boat. So we've really honed the technique to become very good at it, but it's not something you want to go out there and just do without a little bit of experience beforehand.
Yeah, I don't think I'm going to be going out tomorrow to try to tag a great white shark, but I don't know. Who knows what my plans are for tomorrow?
But I hope you get to do it someday.
Oh yeah, for sure. As many times as I've pictured myself up there, like when I see you or someone else, I'm trying to manifest it. I really, really am. But I might not start out with a great white. I might start a little bit smaller. Just like a little bit smaller.
Oh, cool. Yeah, you gotta do it.
And you've been mentioning all these other types of sharks that you've worked with, which honestly I didn't even know about. And it's incredible. The list just keeps going. I really just thought you were the great white guy. You were just the big top dog of the great white shark world. But you've been mentioning all these other species, so is like the Atlantic white shark your main focus, and then you just kind of happen to work with other sharks in the process of working with the Atlantic white shark?
Yeah, in recent years, white sharks have been my primary focus. Even though at any given time we've got a number of projects going on, we're tagging. We're working with the University of Massachusetts Amherst. We're tagging hammerheads in the Florida Keys. We're working on tiger sharks in the Virgin Islands. We've got basking shark work we've been doing for years. And
that's so cool.
Working with the New England Aquarium on sandbar sharks, off
Wow.
Cape Cod in the islands.
Wow.
And so, there's always shark projects happening. But for the last decade, a primary focus of ours has been white sharks off Cape Cod.
And great whites are found pretty much globally. I mean, they're found in the Atlantic, they're found off South Africa, they're found in Australia. Is there a difference between all these different populations of great white sharks? And is there any, population immigration? Like one shark from the Atlantic might hop over to the South African population. Does that ever happen?
Well, genetically, there are very subtle differences. It's all one species, Carcharodon carcharias.
I'm glad he said the scientific name because I would have totally butchered it. Maybe next time I'll say it, but this time I thought I'd let the professional do what the professional does, which is pronounce words correctly. Okay.
From a molecular point of view, there are very subtle differences between the Eastern Pacific white sharks, the Atlantic white sharks, the Mediterranean white sharks, the Indo-Pacific white sharks. There is not a lot of connectivity between these populations.
Okay.
For example, of all the sharks we've tagged in the Atlantic, although a couple of them have crossed into the eastern side of the Atlantic, None have gone into the Mediterranean to mix with those white sharks. There have been a lot of white sharks tagged in the Eastern Pacific, but none have gone to the Western Pacific. But we did have one white shark tagged off South Africa move to Australia. So that was clearly a transoceanic movement. And so there is what seems to be limited connectivity between the Australian, New Zealand white sharks and those in South Africa. But we don't see a lot of transoceanic movement. But there is some. So that allows for what we think, some genetic exchange between those populations.
Gotcha.
But for the most part, populations tend to be isolated.
That's so interesting. And something that I've thought about too is that, if they're separated for so long, I guess this might be on the order of millions or at least thousands of years, but if they're separated and just have a very limited genetic input through a shark that is willing to travel thousands of miles across an ocean basin to join another group of great whites, I wonder if the different populations of great whites might eventually evolve to become, different species, or to where they couldn't interbreed. I don't know, just something I always think about. And another thing that I've heard a lot is that the health of the ocean is dependent on the health of the sharks. And I've heard that in regards to just great white sharks. I've heard that in regards to all sharks. But I guess for you, since you're fluent in white shark, how is the health of the white shark indicative of the health of our oceans?
Yeah, in general, there's been a number of models proposed relative to ecosystem balance. And, every animal has a role within the ecosystem. To remove one of those pieces of the puzzle, you don't have a whole puzzle, right? And so it can lead to disruptions. So when you talk about mesopredators or top predators like sharks, you certainly want to make sure that you have healthy populations of those animals for a balanced ecosystem. If we look at the white shark in particular, and I think that, if we were to talk specifically about the Western North Atlantic white shark population, what we've seen over the course of the last 4+ decades, so 40+ years, is we've seen dramatic measures taken to protect some of these top predators. And a good example of that would be seal populations. So we have seen, seal populations off the northeastern U.S. and Canada driven to the brink of extinction. And then with the passage of the Marine Mammal Protection Act in the U.S. in 1972, we've now seen almost 50 years of protection put forth.
That's awesome.
And we've seen seal populations come back to really high levels. And I think it's to levels that some people don't like.
Oh, that's interesting.
But now we're seeing the white shark come back. And the top predator of seals is white sharks. And so with white sharks coming back in numbers as a result of protection, and as a result of the seals being close to shore, the white sharks coming in, taking notice and feeding on them, we're going to see the white sharks do their job, So if we didn't have white sharks, we'd see these seal populations explode completely out of control. And now we're with the white shark coming back and hopefully doing its job, we're going to see the seal population brought to a level that it should be at. And so that's a really great example of how the white shark as a top predator, if it's allowed to do its job and if its population is healthy, we will see that mesopredator, the seal, population be put in check.
And with that, something that I've always wondered is, for great whites, I would assume that it takes a long time for them to reproduce, or they reproduce very infrequently just based on their size and their lifespan. I may be wrong, but that's just what I assume. And so, do we know if their population in the Atlantic was low, and then when the seals came back, they just grew, and we're now starting to notice them because there's so many more of them? Or were they located somewhere else? Like, were they located off the coast of North Carolina, and then when they moved up for the summer to the north, they were like, oh my gosh, there are seals here, we gotta stay here. And then they just packed up from the south and they just stayed around Cape Cod. Do we know anything about that?
Right. No, it's a great question, and I get it a lot. First of all, we know from historical data that dating back to, the year 1800, that white sharks have been historically distributed all the way from the Gulf of Mexico to Canada. So we're not seeing any change in its basic distribution.
Okay.
All right.
Gotcha.
What we have been able to tease out of long-term data sets, and this is a paper we published a bunch of years ago, is that it appears that the white shark population on the eastern seaboard of the U.S. was hit pretty hard by the expansion of recreational and commercial fisheries targeting shark species as well as tunas and swordfish. All right. And that primarily occurred in the 70s, 80s, and early 90s. And so white shark was primarily taken not only as targeted by recreational fishermen, and some attribute that to the expansion of recreational fishing of sharks brought about by Jaws, but also a massive expansion in commercial fishing for tunas and swordfish, which put literally hundreds of thousands of hooks in the water targeting those species. And white sharks, like many sharks, were taken as bycatch. And we think that hit the population pretty hard and drove it down pretty hard. In 1997, the U.S. government, followed by many states in the early 2000s, prohibited the retention of white sharks. So now we've got, what amounts to 20 years plus of protection of white sharks that allowed that population to go from low levels and start ramping back up. The reproductive rate, as you suggested, is very low, although we haven't really sorted that out. We think females give birth once every 2 to 3 years to maybe a dozen or so young. And so that's a very low reproductive rate. So like seals, it takes a long time for them to come back. And now we believe they are coming back. They're rebounding to historical levels, and not only rebounding, but now coming close to shore in response to the presence of seals. So we're seeing more white sharks too along our coastline and along the New England coastline.
So that was the remarkable and renowned great white researcher, Dr. Greg Skomal. Greg said he doesn't do social media because it can definitely be toxic sometimes, but he does encourage people to follow the Atlantic White Shark Conservancy, which Greg actually heads up himself. The Atlantic White Shark Conservancy supports scientific research, improves public safety, and educates the community to inspire white shark conservation, which, as Greg and I just talked about, is essential for healthy oceans and consequently humans like you, me, and also your cat listening with you that eats tuna. They can be found online at atlanticwhiteshark.org, on Facebook and YouTube at Atlantic White Shark Conservancy, and on Instagram and Twitter at @A_whiteshark. You can support this podcast by becoming an official Elasmite donor at patreon.com/elasmuspod. And again, you have no idea how much it means to me that you give. It's incredible, and I'm so forever grateful. And please rate and review this podcast and send that rating and review to me at elasmuspod@gmail.com to be entered into a drawing, and share this podcast on social media to get 2 extra entries into that drawing. You can find us on Instagram and Twitter at elasmuspod, and on Facebook and YouTube at the Elasmos Podcast, A Shark's Universe. And don't forget to email your marine adventure stories to me at elasmospod@gmail.com so your story can be told on our listeners' episodes. Our website is elasmospod.com, where you can find links to everything, and it was craftily created by Paul McNeely. The theme music was created and performed by Connor Blake and Wes McNeely. Wes can be found on Instagram at wes.mcneely1 and on YouTube at Wes McNeely. I can be found on Instagram at smcneely4335. And now that you've listened to the credits like I've promised, here's my weekly update. I got the shit pinched out of me by a blue crab this week. Good God, it hurts so freaking bad. And yes, it did break skin. But it's different from like a cut, A cut stings because it's quick, but crabs just squeeze. And they don't let go either. It was on my middle finger for what felt like 10 minutes, but it was honestly probably just 10 seconds. But you can't just pull it off either. They usually let go if they're hanging by their own weight and gravity does the job, but this fellow was adamant about teaching me a lesson or something because I was just shaking my hand like a cartoon character. It finally fell off, and right after that, a guy walks out of the crab house with a pair of tongs and says, "Well, that was fun to watch," and hands them to me. I literally just facepalmed right there. Just classic. And within 5 minutes of getting back to the office, we had 5 sets of tongs ordered and on their way. But now, I will say I'm over my fear of ghost crabs, like I talked about a few episodes back, because I know, they can't hurt anywhere close to as bad as that 6-inch blue crab. That's something I don't want to feel again, let me tell you. Anyways, thanks for listening, everyone. Have a great week, and don't get pinched. I'll see you back here next week for an encore episode with Greg on the Elasmos podcast. Later, skaters.